So welcome everyone for the Singapore AI Technology Meetup. So every month we are having this monthly meetup with partnership with organizations across APAC region. So this month we have Algro from Hong Kong. They are a Hong Kong-based startup. They are into unleashing power of artificial cell and gene therapy. So it's a lot of innovation technology that they are working on. So Joel joining us today from Algro, and he will be sharing us about the company, the startup, and how the journey looks like, and also the product journey, as well as how it can be relevant in the innovation landscape. So in our meetup, we are planning to bring a lot of innovative startups every month. So we are having this in both physical and online format. So this month we are having as online format. Next year we are again planning some physical kind of meetups as well. So we have close to 1,000 members in our meetup group, meetup channel as well. So thank you all for joining the meetup. Kindly mute your mic during the session. And if you have any questions during the session, you can ask Joel. So I will hand over to Joel to continue from this point onwards. So hello, Joel. I'd like to hand over to you.
All right. Thank you. Yeah, hello, everyone. How you go on? And let me just get my screen shared over here. Let me see.
Yeah, you can share the screen.
All right. So hopefully, I think you can all see my share screen. All right,
Yeah.
lovely. So yeah, so thank you for the very warm welcome. And yeah, I'm from Allegro, and we are a biotech based in Hong Kong, working on basically new solutions for cell therapy using biomaterials. So our slogan is artificial approach, natural solutions. And throughout this sharing, we're going to explain, besides our product being something kind of man-made, artificial, hopefully we can also tap into basically the emerging field of AI and use artificial intelligence to inform our decisions as well. So first thing first, allow me to introduce myself a little bit more. So I'm Joel, and I'm the Global Alliance Team Lead here at Allegro. And actually, we're just a very small team that started not too long ago. And we have our CEO, Lawrence, up here, and then with Melody. And we also actually have Freeze in the audience with us today, and she's our R&D science lead. And maybe I'll also pull your attention to one of our advisors actually on the board, one of our advisors here actually, so Professor Ying Chao. So she runs the biomaterials lab at the Hong Kong University of Science and Technology. And basically a lot of the research that we tap into, the technology we tap into here is actually come out of research from this lab here. And actually all Lawrence, Melody, Fariza, they all worked at the same lab. They were all students of the same lab here. So that's how it all started. And but before I get into our very heavy tech stuff, might I just ask, maybe just a show of hands of how many of you actually heard of cell therapy before? If you'd be so kind, you can just raise a hand or even just say yes in the chat as well to get a sense of how you all how's your experience or with cell therapy. Anyone? You can take a look.
We have one.
All right. Wait. Oh yeah. Okay, Sean. Thank you very much. So don't worry. So we will get into quite deep into our technology today as well, actually, because in order to explain how AI can come into all of this. But if any points that science get a bit too much, just drop a question in the chat. I'll do my best to answer. So first, what is cell therapy? So basically, it's training your own immune cells to fight cancer cells inside your own body. And so far as it's been very promising new kind of area of medicine that can bring basically cures in patients that never could dream of before. And as you can see, so we have this lady here who has been cancer-free for 10 years using cell therapy. And still, up till very recently, we're still discovering new types of therapies in this field. But there's always a but here, is that cell therapy is actually still very inaccessible to a lot of people because it's just too expensive. And part of the reason why the cost is so high is the manufacturing process. So if we look at the panel on the bottom left here, this is the whole manufacturing process. So first, to have cell therapy, you need to basically take blood samples from the patients, and from the blood samples, you get isolate a special type of immune cells from the blood, and then you do some reprogramming with them, and then comes this activation and expansion part, which is the bottleneck in production. So to have enough cells to inject back in the body, we got to do something called activation and expansion. And this takes a lot of time. You can see 2 to 3 weeks. And this is and in very controlled conditions, in sterile, very clean conditions, because ultimately you're injecting all of this back inside the human body, so it has to be very clean. That means the whole thing has to occur inside a clean room, and the people working on this, as you can see in this photo here, is all gowned up from head to toe, to make sure there's no contamination. And obviously, the people doing this are also very highly trained, so the labor cost is also very high. So because of these reasons, it's actually very expensive to make cell therapy, and particularly this activation expansion part. So if there's a way to kind of supercharge this process, that would be great. And that's exactly what we're trying to do. So let me just, yeah. So we actually have a reagent product which can help in this part. So basically we can produce these therapeutic cells in a better, faster way. But this is actually not our end goal here, actually. We're actually setting our sights a little bit even higher, is that why train the cells, outside the body when you can actually, by a slight modification to our product, inject it directly inside the body and train the cells inside the body to fight off cancer or other diseases. Obviously, injecting in the body, that becomes a therapeutic product, and that's a way longer road ahead for us. So we're starting with reagents first. So so let me just go over some of what's out there in the cell therapy market. So right now, the more established players are CAR T, if you heard about it, T-cell receptor, or TILs, TIL, tumor infiltrating lymphocytes, and also a little bit more up and coming regulatory T cells. Don't worry if you don't know these names. It's not too important, but I'm just setting ground of what type of therapies are out there. So we're just starting with our reagents with these types first, because these are already kind of established out there. We want to basically get our product out to these type of users first. But now we're going to take a little bit more of deep dive into exactly how this activation expansion project works. So we'll have to take a step back and look at how usually immune cells are expanded or activated inside our body, the natural way, basically. So if we look at the left panel here, it looks like a very complex, horrifying diagram, but I'm going to walk you through this. So on the top here, you have something you can see here. It says APC. This is an antigen presenting cell. And basically, this basically approaches the T cell on the bottom here. This is the cell that we are targeting to expand and activate. So when they get close together, the APC and the T cell, they would have on the surface of their cells small molecules called proteins, very, very small things, and they will interact with each other. Basically, the APC is like passing multiple signals actually through these molecules on the surface. And then the T cell, after receiving these signals, they'll be activated. And then they would know, okay, now I have to go after this bacteria, this disease, or in our case, what we want to do is go after cancer. So we've been actually basically the whole scientific community has been able to basically isolate some of these signals outside the body and do it in the lab, basically. So on the right here, you can see the purple here is called activate DC. So it's activate dendritic cell, and this is playing the role of the APC, the antigen presenting cell. And it's activating, again, a T cell on the right, this red cell over here. So you can see there are three main types of signals we're presenting. So we have the antigen-specific interactions, and then you have the basically the synergistic signals on the surface as well. And actually, the activated DC cell also releases something from inside the cell, and then it kind of these kind of IL-12, these small molecules float around, and then when they get picked up by the red T cell over here, the T cell also knows, okay, now I should be activated. I'm going to start going after bacteria, or again, in our case, cancer. And there are actually in the whole scientific community, there has been efforts to basically replace this purple activated DC or the antigen presenting cell in general, so that we can see on the next slide. So I think one of the main ways people are trying to do this is they use a very, very small, so a 4.5 micron or even smaller, a very small piece of bead, like a very small ball, basically. And then they glue or attach these signals on the surface. So there are two main signals, SE28 or SE3, if you want to know. And when these kind of little molecules stuck on the surface of this ball, these two molecules interact with T cells. T cells will, again, would get activated, and then they would start to go after the cancer cells. Another approach other scientists have been using is basically in the vessel that cultures that's growing the T cells on the bottom of the vessel. They also attach these small signals on bottom, and then when the T cells interact with it or get in touch with it, they also get activated. But ultimately, if we look at what the industry is using right now, we can boil down to two main types of ways to do this. So we have, again, the bead that I just mentioned, and this is actually, when it comes to manufacturing, it's kind of like the gold standard. And the reason is because this is actually quite easy to control. And because it's a bead, and you can just add it to the solution to grow the cells. But on the other side, there is something called the feeder cell platform. So this is basically the activated DC dendritic cell. It's the same thing. So on the cell surface, you have these surface molecules, surface signals to interact with the T cells. So both of these approaches have their pros and cons. So when it comes with the feeder cell platform, the good thing about this is as close as nature as we can doing the activation. And because of they have this membrane so all the cells have like this fluid membrane and the soft membrane, enclosing the cell, the T cells that interact with it will know that actually this is, very close. This is home, this is how it's supposed to be. And the interactions really well. But the drawback from using feeder cell platforms is, well, it's because it's a cell, it's alive. So, they can die off during the process. It also is more difficult to manipulate them because you have to feed them as well. So that's why in the industry, they prefer more controllable bead platform. But there are also drawbacks in this bead platform, and that's because first, because the signals are actually just glued fixed on one spot on the sphere, on the ball. So they're not moving around. So this is a little bit unlike how actually, the T cells would interact with them. They're fixed in one spot. They can't move, float around on the surface. And the other thing is because it's a bead, it's hard, it's rigid, it's stiff. So when the T cells come in contact with them, they'll actually touch it, and then they will know that this is actually not really the real deal, because it's supposed to be soft and a little bit pliable, perhaps even. So that's where our product comes in, the Angel platform. So we're trying to have the best of both worlds. So obviously we're a reagent, so it's not something that's alive. And so you can see we have this bigger, like a slightly larger, also is like still a bead, but our bead is actually soft. It's made from hydrogel. I'm going to get into this in a bit. And and we also have this fluid, basically it's very similar to a cell membrane wrapped around it. So that, again, that's moving. And on this moving layer, you have these signals attached to it, which again moves around the membrane. And excuse me, just one sec. All right. And because of this, we get as close to nature as we can while still being a little bit artificial. So, and so let's take a little deeper look into how we make our AimGel basically. So we have a hydrogel. So hydrogel is just a fancy name for maybe like a jelly. Basically, you can think of it as a jelly. And like jelly, actually, we also use some special sugars to make it. So we put it through like maybe a very small hole, and then it gets extruded and becomes this very tiny hydrogel particle here. And already inside, you can load some molecules inside that when the whole hydrogel kind of dissolves, it'll be released. But we don't stop there. We then kind of coat the surface of this hydrogel with special anchors, and with these anchors, we can add our fluid membrane around it, which is very important because we want it to be fluid. And then ultimately, we attach these surface signals on top of the fluid, on the fluid membrane so that they can move around and interact with the T cells. So, and here's just a whole list, a whole host of basically, patents and also journal publications. If you really want to dig deeper into our tech, you're welcome to look at this. But I'm going to explain a little bit more on how tech works anyways on the upcoming slide. So, and this is actually a very neat part. So this explains how do we know actually that our hydrogel surface is actually fluid and movable? Because it's so small. How do you know that it's actually moving? Do you poke it with something? How do we tell? So this is actually an experiment that we did to show this. So what we did is we attach something that's glow in the dark, something fluorescent onto the surface, onto this mobile surface. And then these are actually photos under a microscope. And when we shine a special type of light on it to see this kind of glowing in the dark feature, this fluorescence. And what we do is shine a super strong light or like a laser on it. And this kind of laser would burn off whatever fluorescence, whatever glow in the dark abilities of a particular part of the cell, like maybe not the cell of an angel particle over here. And if it's actually moving, the whole surface is fluid, then basically once it will lose the fluorescence, and then because it's fluid, the fluorescence from other parts of the surface would actually fill in and then regenerate this kind of fluorescence. So let's take a look at actually how, how our angel particles actually grow with the cells actually. So you can see compared to the standard beads out there, when the cells interact with our angel beads, they're actually quite they just love crowding around it because it's very biosimilar. It's like they're tricked into thinking that it's a real cell. So we're going to get into a little bit more specifics on how performance, and then I know people are anxious to learn more about how AI comes into play, but I'm afraid I just have to dig a little bit deeper into how the tech works so that we know how we can pull AI into this. So why Angel first? Why use our product? So one of the main advantages that we have is that during the growth phase, the bottleneck basically, cells that interact with our stuff, Angel compared to the beads can grow faster. And also, and this is another key thing is, and why it's important that we get as close to the natural way as possible is that if you use the beads after kind of adding it to the T cells once, they would grow and expand. But if you add it a second time, actually cells kind of stop responding to it because it's kind of like, oh, you tricked me once already, I'm not going to do it again. But with our Angel, we're actually able to do this again because we can actually still trick them saying, actually, even though we're not really a cell, it will still can view us as something like a cell. And then activate. And here we actually look at the more nitty-gritty detail of the performance of our Angel. So basically, we're able, when the T cells expand, actually there are they grow into different subtypes of T cells. So some subtypes are actually more useful than others when it comes to cell therapy. So with our technology, we're able to grow a specific subtype, we call it memory T cells, to a higher percentage, basically. And not only this, if we change the type of signals on the surface, we're actually able to kind of dictate, control which subset of T cells gets grown, basically. So that's actually very important. This feeds directly into how AI comes into play. So maybe I'm just gonna skip ahead a little bit. So right now, so where to show where we are at the cell therapy market, we're actually right in the midstream. So we need upstream raw materials from other suppliers, like the protein, the signal molecules on the surface, and also obviously the special type of sugars that we use. And then we make our AimGel, and then we sell it to downstream producers of cell therapy and also people who do research in cell therapy, like biotechs or universities, research institutions. And and this is only on the reagent side. So we also mentioned something about doing this right inside the body, in vivo, doing immunogenic response right inside the body. So I'm just going to go a little bit very briefly over this. So to explain the science behind it, whenever you inject something inside the body, usually the body doesn't like it, because it's actually quite dangerous to have foreign stuff inside you. So in our own immune system, actually, there's a special type of cells called macrophages that would attack anything that comes in from the outside. That's just called foreign body response. And because of how we make Angel, actually, and because it's very kind of biocompatible, very lifelike, we're able to suppress this reaction. So once we inject it into, in this case, still mice, they're able to stop macrophages from basically attacking them and kicking them out. But still, they can activate a particular part of the immune system called dendritic cells, which we mentioned before, to activate other, the immune, the T cells and all that. So at least in the mouse experiments, we can show this. So I'm not going to go too detailed into this rabbit hole for now. And so basically, we're pushing out our reagent product this year actually, and so we're just working on the conventional T cells and also tumor infiltrating lymphocytes. And then every year we're targeting new cell types, so regulatory T cells, natural killer cells, gamma delta T cells. These are the reagents, and then we have the macrophages and the T cells for the in-body stuff. So we're very young startup, and we're very blessed to have gone really far. But I'm gonna just move ahead and go into how does AI come into this. So we went through a lot of deep tech already, but how does AI feed into this? So what does AI do? So we need to use AI to find the right signals for this right cell type, basically. So whenever our angel interact with the immune cell, basically we have something on the surface on the angel, and there's also something inside angel being released to interact with the immune cell. And right now, obviously, the industry is using two particular signals, but already within our own testing, we know that while these two signals work, there are also other signals that also work. And and with the signals, all of them are synergistic. So 1+1 is more than 2 always. And some of them actually are antagonistic, so they work against each other. And given how complex the human biology is, the number of signals that we can tap into is actually almost endless. It's this crazy combination. So we need to look at which type of signals. There's way more than 5. And then for each signal, we need to look at how many to use. What's the concentration? And ultimately, we need to find the balance of combinations that to use. So we already in our own in-house research, we've tried 2 to 3 combinations of signals of again, 4 to 5 even. So we need to feed all this information basically into an AI model to basically look at how different signals can lead to the growth and expansion of different cells, T cells to dendritic cells to macrophages. And basically, if we look in take a look at how to set up the experiments and also building on the model. So for now, our independent variables are signal type, signal concentration, and combinations that we just went through before. And what we can get out through our experiment is how fast it can grow, and then the type of cells that grow out of a particular type of signal. And we can also analyze by scrutinize basically by the cells that grow what's on the surface as well. That also gives a good indication of what we can do, at least of what the signals do. And because we just really started on this, we're still doing very kind of human intensive, labor intensive experiments. So we don't have that much data points, so like less than 100. So we know in terms of AI, this is nowhere near enough. And basically, we have just different combinations of signals, and then we see how well the cells grow after this. And to maybe even take a step back from this, so this is purely experimental view on this. We can actually look at the structures of these molecules on the surface, and then we look at what works and what doesn't work. And perhaps by analyzing these structures and then comparing it with what works and what doesn't work, then we can still basically kind of work our way backwards and see actually what type of signals is actually have potential to activate the different type of immune cells out there. So this also means that we will be looking at the whole genome. So basically the genomes are DNA. So basically that's the blueprint on how proteins are made, so how the signals are made. And the proteome is basically how, the whole collection of proteins that we can express on the surface. And basically we're just dipping our toes in AI. So we're still looking for ways to build a model, basically. From a very initial understanding of what AI is and what AI can do, because we don't have very distinct, inputs output features we can actually tap into. We're thinking of starting with unsupervised learning first to look for clusters so that we have, a pathway at least forwards, and then from that we can fade into the supervised learning model. But again, this is very preliminary, and we actually need quite a lot of effort just to generate enough experimental results to get us started. So maybe we can go over a little bit of how we plan to go ahead and with AI. So basically in the coming year, we're just trying to build up our AI capabilities. If any of you, are interested as well, we're happy to hear from you. And obviously we need a lot of data to feed into AI. So we're going to be doing quite a lot of experimentation just to get enough data. And then the year after, we're going to start training our models. And because it's always hard to self-generate data, hopefully we can work with other partners to actually generate even more data to feed into our models. And ultimately, hopefully in 3 years' time, we can take the learnings that we have from our AI model and start incorporating designs, basically what kind of surface signals we can put on the surface of our AimGel product, so that we can really supercharge this cell therapy manufacturing process. So that's actually a breeze through a little bit quicker. So that's the summary of what we do. We have in our AimGel platform, and actually how we plan to basically leverage AI to basically supercharge cell therapy manufacturing, using our product and using AI to inform how we design our product actually. So with that, I'm very happy to answer any type of questions out there from the audience. Uh, I did breeze through a little bit of tech because maybe I don't want to bore you too much, but if any case there are parts that you feel left unexplained, feel free to ask me in the chat as well on how based on how our technology works and how or I asked how AI can feed into this model, in a more elaborative way.
There's one question in the chat, interested in how AI can help in cell therapy. I think some part you have already
Oh yeah,
answered.
so yeah, so how AI can help in cell therapy. So there are actually, I think, many different angles you can attack this. So from our perspective, from our immediate perspective at least, we'll be looking at how to find the right combination of basically signals to interact with the cells of the immune cells to really kind of grow and supercharge them. But there are also other angles as well, basically. So uh, to kind of guess which cell types work or what doesn't work, uh, you can also model this with computer models, and you can also use AI to do this as well, actually. But that's not something that we have the capabilities or at all at this stage. We're still very focused on our product. So, but we've seen the literature out there. There are other researchers, not necessarily commercial, they're purely academic. They're looking at ways to basically screen through the whole, genome, or as I mentioned here, or the proteome. So basically, look at all the existing, um, proteins out there that can be on the surface of the cell and see how they could be related to this activation expansion process during cell therapy manufacturing. So I don't think the scientific community is really tapping the full potential of this yet, because AI just really started, came back in resurgence basically this year and the year before, and basically all the industries, including science included, life science included, were just starting to kind of come to grasp with how powerful it can be, what a helpful tool it can be. So that's, I think, that answers how in a very specific way on what we do, how it can help, and also maybe in a broader way and across the whole industry, how this can work. And maybe also digging a little bit deeper. So right now, cell therapy is actually divided into two kind of approaches. One is something called autologous. So that's using the immune cells using from the same patients, and then training them, activating and expanding them and injecting back in the body. So that's autologous. So you use the same cells from patient, but there's also something called allogenic. So that means it's this universal donor uh, that you take the blood from and then isolate this donor's cells, and then the cells from this donor can basically uh, cure or can be injected into all different patients out there. So obviously, the allogenic way, the second way, is actually it's sometimes more difficult because you don't know how to fine tune it in a way that you can actually make it work for everybody. So perhaps AI can also help in optimizing this as well. Look for factors, look for cues to make allogenic cell therapy products more adaptable, for everybody basically. And this is actually another reason why cell therapy is also very expensive, because it's right now a lot of cases done case by case. You have a patient here with leukemia, and then you make the cell therapy products for this person specifically, doing autologous cell therapy, and and the next person comes along, and then you start process all over again, with and this obviously increases the cost. But with allogenic, you have this universal donor, making the therapy. Then basically you can have patient A, B, C, D, all the way down to Z, and basically they're just using the same uh, cell therapy product from the same universal donor, so you don't need to start the process from scratch again. But developing allogenic therapy is actually more difficult, and hopefully AI can also help in this regard.
If you have any other questions, you can post in the chat as well.
Yeah.
I have also another follow-up question. Like,
Oh yeah.
was your like the experiment process the average time and like reiteration time? Like,
That's a very uh let me see if it's a long time, because so you're dealing with cells growing. So we're talking at least weeks for the experiments, given these are more complex cells. Because if you play with bacteria, it's actually, they replicate very quickly. 20 minutes, E. coli, your double population. With immune cells, obviously, it's a bit slower. So from start to finish, I think we're talking at least weeks. Um, so right now we're doing this manually. So it's still very, very, very tough actually uh, for freeze actually. And so we're also, because of this, we're also looking into automation as well. Uh, how we can tap into batch processing so that we can do a lot of experiments in one go. And hopefully, and with AI, we're also hoping to tap into how we can better design our experiments. Because let's say we keep it very simple. We want to play with a combination of three different signals. And we're just playing with the concentrations, basically. How many experiments do we need to do? How many sub experiments do we need to do? So do we do just a normal 1-to-1-to-1 split, and then we just iterate until 1-to-1-to-2 ratio, and then 1-to-1-to-3, and then we do 1-to-2-to-2, and how, and it will be very tough on us to try all the combinations out there. So one of the reasons we're really anxious and hopeful to get into AI is to basically, if we can just kind of narrow down and reduce the number of experiment subsets that we have to do. So maybe we can just do with 1-to-1, and then we'll try maybe 1-to-1-to-10 for different, and 1-to-10-to-1 and 10-to-1-to-1, and then try different combinations. And already AI can take that kind of information and then generate actual insights to see actually the most optimal combination for the first variable is actually 2.5, 3 something maybe. So we're so that's how immediately I can feed into this. Like if we don't look at all the signals out there, we're just sticking to, let's say, just 3, then this is how AI is going to work for us. So so yeah, we're really looking forward to how we can, what we can do here with AI, and we're very hopeful and optimistic, but it's still a long road ahead for us in this regard.
yeah, of course. Any other follow-up messages or any questions you can ask in the chat? Otherwise, we can wrap up for the today's session. If you have any questions about today's meetup, you can ask. Seems like I think no more questions. You can wrap up the finalize, like you want to like have the final word.
Oh yeah, sure. Yeah, the final. So yeah, thank you for joining today. We're just really excited to be in this space. We just raised our company last year. The tech is really fresh, straight out of the lab. We're really looking forward to having an impact in the industry. We know it, when it comes to biotech, it's always a long road. But we have faith in our team and our product, our designs, and hopefully, and we can make an impact in this space, because cell therapy is still kind of this high-end medicine, personalized medicine out there, and we do want to get to the masses. It shouldn't be priced out for basically most of the world. So hopefully, with our technology, supercharging with AI, we can really make a difference. So you can check out our website if you want to learn more. And yeah, keep an eye out for us. And if you have, you know, you're looking for collaborations in the AI space as well, just I think there's you can just drop me a message on LinkedIn as well. Uh, and yeah, so or you can find us on our website, on Twitter, all that. So yeah, really we're lucky to be in this space really for us, and hopefully uh, we can really make a real impact. So yeah, thank you. Thanks for having us, Yudita.
Yeah, of course. Yeah, welcome,
Bye.
Joel. Yeah, so thank you, Joel, for your time and taking the time to talk about the startup and the cell therapy and the innovations behind that. And like, we are looking forward to have you again in upcoming session next year as well. So we are like every month we are planning these sessions online and physical format. So thank you. I would like to thank Joel again for having your time and like taking the effort to showcasing the innovation around cell therapy. And also like to
No
thank
problem.
everyone who joined to today's meetup as well. So thank you everyone for joining the meetup. So keep engaged to our meetup channel to look for the upcoming session. And the session recording will be also shared in the meetup page. So you can also access this recording, and any clarification you can contact Joel as well. And thank you everyone. Have a good evening.
All right, thanks. Yeah.
Okay.
Hope you all have a lovely day.
Thank you.